In this video lecture, we will be discussing the different senses through which we get information about the external world. To make sense of the external world, we need to both get information about things in the world and also then to organize, interpret, and experience that information. Sensation refers to the processes by which this information about different things in the world is obtained through our sensory receptors and then transmitted to the nervous system to be processed, organized, interpreted, and experienced. There are different ways of describing, defining, and classifying our different senses. People have proposed that we have anywhere from 5 to up to 53 different senses. For our purposes, we are going to discuss the senses broadly divided into two major categories. Our first major category of senses are our interoceptive senses. Our interoceptive senses are the senses that are responding to information internal to the body. So they are receiving information from internal sensory systems. These include senses like our vestibular system, our sense of our position and motion through space. These are also senses that give us information about internal bodily sensations that lead to sensations such as feeling hungry, feeling thirsty, feeling as though we need to breathe, or experiencing nausea. In contrast, our exteroceptive senses are providing us with information about things outside of the body. So our exteroceptive senses are responding to external sensory systems. These include the 5 senses that you are probably familiar with, things like our sense of touch, our sense of taste, our sense of smell, our sense of hearing, and our sense of sight. We're going to begin by discussing some of our interoceptive senses. Remember that these are our senses that are responding to internal sensory systems and providing us with information about things that are happening inside of the body. One important interoceptive sense is our vestibular system. The vestibular system is responsible for our ability to maintain our balance and our body's posture. The sensory receptors for the vestibular system are located in the inner ear. So if I had asked you where your sensory organs are located, you might have guessed that our sensory organs include things like the tongue, our eyes, our ears. You might have even said your skin. The sensory organ for the vestibular system is located in the inner ear, and these sensory receptors respond to movements of the head and gravity to give us information about our body's posture and help us maintain our sense of balance. Another important interoceptive sense is our sense of proprioception. Our sense of proprioception gives us information about our body's position in space. Along with our sense of proprioception, we also have our sense of kinesthesia. This is our ability to perceive not only our body's position at a static moment in time, but our ability to perceive the movement of our body through space. One way that we can learn about our different senses is, similar to our discussion of the functions of different brain regions, we can discuss what happens when there are disturbances to our different sensory systems. So we're going to talk briefly about a disorder of the vestibular sense called benign paroxysmal positional vertigo. Benign paroxysmal positional vertigo occurs when an individual has symptoms of vertigo that are triggered by movement or positional changes. Vertigo refers to experiencing a false sense of motion, so feeling like you are moving when you are not actually moving. So benign paroxysmal positional vertigo occurs when an individual feels like they are moving after changing position or moving part of the body. So for example, somebody might turn their head to the left and suddenly experience these unpleasant senses of vertigo. Or if they change positions, for example, going from lying down to sitting up or going from sitting to standing, they might experience this unpleasant sense of vertigo. And the cause of benign paroxysmal positional vertigo is really interesting and has to do with those sensory receptors that are located in the inner ear. Benign paroxysmal positional vertigo occurs when calcium carbonate crystals are dislodged from one of these organs called the utricule and migrate to the semicircular canal. So these calcium carbonate crystals begin sending signals to the brain that movement is occurring, even though movement is not actually occurring. So the treatment for benign paroxysmal positional vertigo is a really interesting treatment that is a physical maneuver that is designed to dislodge these crystals and to move them to a correct position so that they stop providing this incorrect sensory input. So we're going to watch a short video clip of an animation that will show this Epley maneuver that will both show how these crystals are producing this false information that is leading to the sense of vertigo, and also how this series of motions will move these calcium carbonate crystals in the semicircular canal and get them back into the correct position to end these symptoms.
Normally, only with head movement does fluid within the inner ear also move, informing the brain that a head turn occurred. However, should a crystal called an otolith gets displaced into the posterior canal of the inner ear, BPPV dizziness occurs. Fluid movement occurs due to the crystal rather than head turns, causing the brain to think movement has occurred even though none has happened. The Epley maneuver is performed to treat this posterior canal BPPV by trying to get the loose crystal out of the canal. This maneuver is started by turning the head 45 degrees and the body laid back such that the head is extended about 20 to 30 degrees. If BPPV is present, eye twitching called nystagmus will occur. After 30 to 60 seconds, the head is turned 90 degrees to the opposite side and held for another 30 to 60 seconds. The head is then further rotated another 90 degrees, accomplished by having the patient turn onto their side, which is then held for another 30 to 60 seconds. At this point, the head should be turned 45 degrees from the horizontal. When performing this maneuver, another individual can help with these position changes. The maneuver is then completed by having the patient sit up. So what's going on with these position changes? Essentially, the head is moved in such a way to manipulate the crystal to fall towards the canal opening. With each position change, it takes about 30 to 60 seconds for the crystal to settle into the most dependent position in the canal. If turns are made before the crystal has a chance to settle, the crystal may fall back the wrong way and the maneuver will fail. It is also important that the head positions are angled correctly, or else the crystal will not settle to the correct position in the canal and the maneuver will fail. Also, if the position changes are done too slowly, the crystal may not settle quickly enough from lack of momentum and the maneuver will also fail. Once the crystal falls out of the canal, the dizziness should resolve if due to posterior canal BPPV.
Next, we're going to discuss our exteroceptive senses. So remember that these are our senses that are responding to external sensory systems and giving us information from outside the body. These include things like our sense of touch, our sense of taste, our sense of smell, our ability to hear, and our ability to see. We're going to start by talking about our somatic senses. Our somatic senses are going to be familiar because we discussed these senses when we discussed the somatosensory cortex. So these are senses that are providing that input to that somatosensory cortex. Our somatic senses have to do with our ability to perceive touch, both things like pressure, but also being able to perceive things like the temperature or being able to perceive pain. One of our somatic senses is haptic perception. This is our sense of touch, and the sensory receptors for haptic perception are distributed throughout the skin. These are called mechanoreceptors, and there are different kinds of mechanoreceptors that are specialized to respond to different kinds of pressure and information from the skin. So some of these mechanoreceptors respond to pressure. They might respond to different intensities of pressure. Some of them respond to low or high frequency vibrations, and some of them respond not to pressure on the skin, but to stretching of the skin. So these different sensory receptors are providing us with different information about where we are experiencing touch and the nature of that sensation. Another somatic sense is our sense of thermoception, and this is our ability to perceive temperature. The sensory receptors for thermoception are called thermoreceptors. These are located not only in the skin, but also in our skeletal muscles, the liver, and the hypothalamus. We have two different kinds of thermoreceptors receptors. Some of these are warm thermoreceptors that provide us with information about temperature that is greater than the temperature of the human body. Other thermoreceptors are cool thermoreceptors, and they give us information about temperature that is less than the temperature of the human body. The final somatic sense that we'll discuss is our sense of nociception. This has to do with our ability to perceive harm or pain, or also perception of potential harm or pain. We are talking about this as an exteroceptive sense and getting information from outside of the body, similar to our sense of touch through the skin. But there are some ways that we perceive pain that we can also think of as more of an interoceptive sense that are giving us information from inside of the body. Our sensory receptors for nociception are also distributed throughout the body. These are called nociceptors. They are found not only in the skin, but they're also present in our muscles, our joints, our bones, and our organs. And these sensory receptors respond to tissue damage. They also respond to signals that indicate the threat of tissue damage. So our somatic senses altogether are giving us information about our sense of touch. The next exteroceptive senses that we're going to talk about are referred to as our chemical senses. And these are called our chemical senses because these are senses where the sensory receptors are responding to the presence of molecules in the environment. Our first chemical sense is our gustatory sense, or our sense of taste. The sensory receptors for our sense of taste are located on the tongue. These are gathered into bundles that we call taste buds. And these different taste buds are responsive to different characteristics of flavors. So there is debate about how many different kinds of taste buds we have and how many different characteristics of things we are able to taste. But we know at minimum that we are able to detect and differentiate 6 different taste characteristics. These include the sweetness, the saltiness, the sourness, the bitterness of things that we are tasting. Another characteristic is something called umami. This is a richness. It's sometimes described as a meaty flavor, even though not everything that is strong in umami is an animal product. And then finally, we also have taste buds that are responsive to the fatty content of food. So our sensory receptors for our gustatory sense transmit information up through the medulla to the thalamus. That information is then sent to the limbic system and goes on to the gustatory cortex. So similar to the other cortices that we talked about our primary auditory cortex for hearing and our primary visual cortex for sight. The primary gustatory cortex is the primary region of the brain that is processing information about taste, and it is located in the parietal lobe. Closely related to our gustatory sense is our sense of olfaction, or our sense of smell. The sensory receptors for our sense of olfaction are located in the mucous membranes of the nose. And an interesting thing about our sense of olfaction is this is the only sense where this incoming sensory information does not go through the thalamus before it is relayed to other regions of the brain. So sensory information from these sensory receptors are transmitted directly to the olfactory bulb. So the olfactory bulb is a place where all of these olfactory nerves converge, and this incoming olfactory information then goes from the olfactory bulb directly to the primary olfactory cortex, which is located in the temporal lobe, and also to the limbic system. The next sense we'll discuss is our sense of audition or hearing. The process of audition is a mechanical amplification process through which sound waves in the environment are converted into fluid waves in the ear, which are then converted into neural impulses that we perceive as meaningful sounds. Sound waves in the environment have different characteristics that correspond with different characteristics of sounds that we perceive. So for example, the wavelength of a sound wave, which has to do with the distance between the peaks or troughs of a sound wave, is related to our perception of the pitch of a sound, so whether a sound is perceived as a high sound or a low sound, and the amplitude of a sound wave, so that is the distance between the peak and the trough of a sound wave, is related to the perceived loudness or volume of the sound. We are going to look at how transduction of sound waves occurs through the different parts of the ear. So to take these sound waves from the environment and turn them into neural impulses that can then be perceived and interpreted in the brain, these sound waves need to be taken from the external world and they need to be turned into an action potential. They need to be turned into a neural impulse. So this begins in the outer ear. First, sound waves in the environment are funneled through the auricle and the pinna into the auditory canal. So those sound waves are funneled into a smaller area. They become magnified. These sound waves travel down through the auditory canal until they reach the eardrum. The eardrum is a thin membrane between the outer ear and the middle ear, and when these sound waves reach the eardrum, they cause the eardrum to vibrate. So this is the point in the transduction of sound waves, where these waves are turning from sound waves in the air into physical vibrations in the ear. The next part of the process occurs in the middle ear. So in the middle ear, these vibrations are going to be transmitted through a series of bones that are called the ossicles. The first ossicle in the middle ear is called the malleus, or the hammer. The malleus is caused to vibrate by the vibration of the eardrum. The malleus vibration then hits the incus or anvil, that is the middle ossicle of the middle ear, and the vibration of the incus is then going to cause the vibration of the third and final ossicle, which is called the stapes or the stirrup. The vibration of the stapes is then going to reach the oval window. Similar to the eardrum, the oval window is a thin membrane between the middle ear and the inner ear or the cochlea. So the vibration of the stapes is going to cause the oval window to vibrate. We then reach the inner ear or the cochlea. The cochlea is this snail-shaped structure in the inner ear, and the cochlea is full of fluid. So when the oval window vibrates, that causes the fluid inside of the cochlea to vibrate. When that fluid vibrates, it causes the basilar membrane, which is a membrane that is lining the outside of the cochlea, to deform. The basilar membrane is lined with hair cells, and these hair cells are our sensory receptors for our sense of hearing. So different characteristics of sound waves in the environment will cause different kinds of deformation of the basilar membrane that will lead to different patterns of activation of the hair cells. So some hair cells respond to sounds at different pitches. So the way that sound causes the cochlear fluid to vibrate, which leads to differences in how the basilar membrane is deformed, determines what hair cells are activated and how they send signals. And then that information is transmitted from those hair cells. Those sensory receptors send information through our auditory nerves to the thalamus. Then that thalamus sends the information to our primary auditory cortex in the temporal lobe. So that is the process of how sound waves in the environment are mechanically amplified and turned into fluid waves in the cochlea, which are then turned into neural impulses that are transmitted to the auditory cortex, where they can be perceived. The final sense that we'll discuss is our sense of vision. This is our ability to see, and similar to hearing, in vision, we are converting waves in the environment into a neural impulse that is then turned into meaningful information about the world. But in the case of vision, rather than converting sound waves, we are converting light waves into neural impulses, which is then turned into meaningful information about the visible world. Similar to sound, the wavelength and the amplitude of light waves from the environment are associated with different characteristics of what we perceive about the external world. So the wavelength of waves of light has to do with our color perception. So long wavelengths are perceived as redder, and short wavelengths are perceived as blue or purpler. And the amplitude of a light wave is related to the intensity of the color that we perceive. We're going to talk about how the transduction of light waves into neural impulses that are then perceived as information about the visible world by discussing how light travels from the external world through the anatomical structures of the eye to produce this neural impulse. So the first part of this process is that light waves bounce off of things in the environment and enter the eye. These light waves first travel through the cornea. The cornea is the transparent outermost layer of the eye. One of its primary functions is to protect the eye. The cornea also starts to focus light waves on the back of the eye onto the retina. The iris is an important structure in the eye. The iris is connected to smooth muscles. It's located between the cornea and the lens. The iris is the colored portion of the eye. And what this does, it controls the size of the pupil of the eye. The pupil of the eye is the small opening in the eye through which these light waves are passing. So because of that, the size of the pupil determines the amount of light that is able to enter the eye. So by controlling the size of the pupil, the iris is controlling how much light is able to enter the eye. And finally, after passing through the pupil, these light waves pass through the lens of the eye. The lens of the eye is a curved transparent structure whose function is to focus the incoming light waves onto the retina, where these light waves will trigger our sensory receptors for vision, our photoreceptors. So once light waves have traveled through the eye, it reaches the retina. The retina of the eye is light-sensitive tissue at the rear of the eye that contains the sensory receptors for our sense of vision. These are called photoreceptors. We have two primary types of photoreceptors on the retina. One type of photoreceptors are called cones. These are photoreceptors that are sensitive to color. Our cones have high acuity, so this means that we are able to perceive fine detail, and our cones are concentrated in the fovea, or the center of our retina. And our cones work best under conditions of bright light. The other type of photoreceptor that we have on the retina are our rods. These are photoreceptors that do not respond to color, but they are sensitive to lightness and darkness. In contrast to our cones, these have relatively lower acuity, so are less sensitive to detail, and they are more concentrated in the periphery of the retina. Also in contrast to the cones, our rods function better in conditions of low light. After these photoreceptors are activated by light waves, the information is transmitted to the optic nerve through a series of cells. So when the rods and cones are activated, they then transmit that nervous impulse to our bipolar cells. So bipolar cells are intermediary cells in our visual system that exist between our photoreceptors, between the rods and cones, and our ganglion cells. And our ganglion cells, the axons of these cells converge to form the optic nerve. That is the nerve through which these neural signals are then transmitted into the brain to the visual cortex. So an interesting thing about the bipolar cells is that rods and cones are connected to different numbers of bipolar cells. So each cone has its own bipolar cell that it transmits information to, and that information is then transmitted to ganglion cells. In contrast, a single bipolar cell might receive input from multiple rods. So this provides an explanation for why we have different levels of visual acuity between our rods and our cones. So because bipolar cells are receiving information from multiple rods, we have less of an ability to differentiate which of the photoreceptors that information is coming from. In contrast, because each cone has its own bipolar cell, we have better acuity because we are better able to determine exactly what photoreceptor was activated and sending that message in. So these photoreceptors are sending information through the bipolar cells to the ganglion cells. The axons of those ganglion cells are converging to form the optic nerve, and the optic nerve is then going to transmit that neural impulse to the brain. The optic nerve is then going to transmit these neural impulses to the brain, where this information can be perceived to result in perceptions and meaningful information about our visual world. The place where the optic nerve exits the eye is called the blind spot. And it's called the blind spot because we have no photoreceptors on this part of the retina. So the optic nerve exits the eye, and this sensory information is then transmitted to the brain, and information from the left and right visual fields is sent to the contralateral hemisphere of the brain. So information from the left visual field is perceived on the right side of the retina of each eye, so both the left and the right eye. The right side of the retina has the photoreceptors that perceive the left visual field, and the axons from those photoreceptors travel, they go out the back of the retina at the blind spot, and these axons then cross at a location called the optic chiasm. So the optic chiasm is the place where the optic nerve crosses over from one side of the body to the other side of the brain. Information from the right visual field of the external world is perceived by photoreceptors on the left side of the retina of both the left and the right eye, and these axons converge and the optic nerve then exits the eye, and again information from the right eye is going to cross the optic chiasm and go over to the left hemisphere of the brain. So information from the left and the right visual fields of the environment are perceived by both eyes, by photoreceptors on the retinas of both eyes. Information from the left visual field is going to be processed in the primary visual cortex of the right hemisphere of the brain, and visual information from the right visual field of the external world is going to be perceived by photoreceptors on the left side of the retina and is going to then be processed by the visual cortex in the left hemisphere of the brain. So similar to all of our exteroceptive senses other than smell, these optic nerves are going to transmit neural impulses that are then going to make their way to the thalamus. The thalamus is then going to transmit this information to the primary visual cortex. This information may also be processed by different specialized regions that process different types of visual information. So for example, there are different structures in the brain that are specialized for processing information about physical objects or about faces or about spatial location. So that wraps up our review of our different senses. We have our interoceptive senses that are responding to sensory systems and information internal to the body, things like our body's sense of balance, our ability to sense where our body is in space, and our ability to sense our body's movement through space. Our interoceptive senses also give us information about internal bodily sensations, such as hunger and thirst. And then we have our exteroceptive senses. These are senses that are giving us information about things that are external to the body. These include things like our somatic senses, our sense of touch and temperature and pain, our chemical senses like our sensation of taste and smell, our auditory sense, our ability to hear, and also our visual sense.